So I'm Dr. Koen Deloose. I'm head of the Department of Vascular Surgery in AZ Sint-Blasius in Dendermonde, Belgium. And I'm also involved with ID3 Medical in research, training, and education on the vascular level. Well, first of all, of course, I really wanted to jump into the real life lesions. So I think the literature is plenty with studies with 5 to 10 centimeter lesions. And I'm not seeing this kind of lesions in my daily practice. So I really want to jump into long and complex lesions. Long lesions, quite clear according to the TASC C classification, it is about TASC C and D, more than 15 centimeter. Complex is a little bit more difficult to define. We have a lot of factors that are dealing with complexity. I'm thinking on multi-level disease. I'm thinking especially on calcium. I'm thinking on the heterogeneity of lesions, the comorbidities of our patients. So these make the lesions really complex. But finally, at the end, I think also besides calcium, the length of the lesion is quite an important factor of complexity, and it's really contributing a lot to a failure of our treatment on the longer run. We know out of a study of SOGA that in determining the risk factors, that length more than 15 centimeter was contributing 2 points out of 7 to the risk factor score for restenosis afterwards. So long lesions, and then in my experiences, what is the best to tackle this kind of long lesions? It is first of all vessel prepping the vessel. How you are doing this, it's up to the physician's discretion. POBA, specialty balloons, atherectomy devices, whatever what is available and reimbursed can be used. A second important pillar is drug elution. And right up to now, when I'm looking at the literature and all the presentations in the world, I notice that still paclitaxel is the most efficient drug to use. I think also the safety debate is a little bit on a lower platform today, and we don't have the durability of other drugs on the longer run. The results, so we need to wait for this. So right up to now, paclitaxel elution. And then last but not least, in complex lesions, we need scaffolds. And then when we are looking at the portfolio of iVascular, we notice that we have 2 very important components over there. We have on one side the Luminor paclitaxel eluting balloon, 3 microgram per square millimeter with an innovative nanotechnology that is creating a very homogeneous coating of our balloon on one side, proven efficacy in the FPAC trial, in the Mirjam trial, in the Luminor registry. And on the other side in the portfolio, we have a very good scaffold. We have the iVolution stent, today available with the iVolution Pro delivery device, what makes the deployment even more easy, more comfortable, more ergonomic, and more precise. So I wanted to see in this trial the combination of these 2 top products in order to tackle long, complex lesions, if this is a winner, and if 1 and 1 is more than 2, is actually 3. This was the rationale of our trial.
Thank you very much. Can you tell us about the design, patient population, and endpoints of the TINTIN trial?
Yes. So the TINTIN trial is a physician-initiated prospective single-arm study. 100 patients are enrolled in this trial. And the most important thing is to test the combination therapy of Luminor on the 018 or 035 platform with the iVolution stent scaffold in lesions, femoropopliteal lesions, TASC C and D. So a minimum of 15 centimeter length is required. It is a single arm. There is no randomizing or comparable arm. 9 Belgian centers were enrolling patients in this trial. And finally, the primary endpoint was in terms of efficacy was the primary patency at 1 year, and the safety primary endpoint was the major adverse event rate at 30 days. I presented these data last year during the LINC symposium, but today in this LINC edition, I want to present you 2 years data. So I want to see also the durability of this treatment in complex lesions on the longer run. 2-year data with freedom from target lesion revascularization, with survival rates, and with major adverse event rates. That's what I'm going to present in this LINC edition. So as we all know, we have, as I presented last year, we had a fantastic primary patency result after 1 year of about 90.5%. But we have today the 24-month data available, and we notice a freedom from TLR at 24 months of 89.4%. I wanted to point out that we are speaking about, first of all, lesions of 24 centimeters in length, so amazingly long lesions. 60% of these lesions were chronic total occlusions, so 60% of occlusions is quite a high number. And we are also dealing with very sick patients. 28% of the patients were CLI, CLTI patients, with 26 of them were really Rutherford 5 patients with non-healing ulcers. So in a very diseased population, in very long and complex lesions, reaching a freedom from TLR at 2 years of almost 90% is for me extremely exciting. Notice this. Another story was the survival rate. The survival rate at 2 years was 81.6%. And in the beginning, when I was confronted with these results, it was rather disappointing. I was jumping more in detail into the causes of the high mortality, and 18.4% after 2 years. First of all, I was thinking, of course, on the COVID pandemic situation, but I noticed that since the beginning of the pandemic, there were only 4 deaths, and these 4 deaths were not related to the COVID-19 situation, but rather on cardiovascular complications. Of course, on a second point, we are thinking on the paclitaxel issue, the safety issue, but also there we jumped into detail and we didn't notice any correlation or causal relationship with, on one side, procedure and devices, and on the other side, the mortality. So I have no clear explanation from this side. So finally, my third explanation for this, and I think this is the real one, as I've already mentioned, we are dealing with extremely sick patients, 1 out of 3 CLTI patients with lesions of 24 centimeters, 60% chronic total occlusions. So a very diseased, cardiovascular disease population, and this is the reason for the rather disappointing survival rates at 2 years. If we are looking at safety outcomes at 2 years, we noticed, as I've already mentioned, in terms of major adverse events, the 17 deaths. We have 9 TLRs, so 4 more compared to the 1 year results. We have no amputation or no thrombosis after 24 months. So these were a little bit the outcomes at 24 months. In terms of clinical improvement, we've noticed that after 1 month and 12 months, there was a tremendous shift from Rutherford 3, 4, and 5 towards 0 and 1. Well, these results are sustained at 24 months. So also from a clinical perspective for our patients, patients are doing very well. I think the take-home messages, at least for me, is first of all that lesion length is a quite important restenotic risk factor predictor. So take care of a durable treatment if we are thinking on this kind of lesions. Durable treatment, in my opinion, is the combination, the three pillars of vessel prep, drug elution, and scaffolding. And this is proven in this Belgian TINTIN trial, where we've noticed that with the combination of the Luminor drug-coated balloon and the iVolution stent, that we are reaching in lesions of 24 centimeters, 60% occlusions, and almost 30% of CLTI patients, a primary patency of 90% at 1 year, and a freedom from TLR of 89.4% at 2 years. When we are benchmarking this combination therapy, DCB with stent, we notice, and I know that it is dangerous to compare trials, but just from an educational and interest point of view, they are doing slightly better than DCB and bailout stenting, and they are at least equivalent with the modern generation of drug-eluting stents. Well, of course, like I've mentioned in the take-home message, we can benchmark this kind of treatment. So the combination therapy of Luminor and iVolution stent in a single arm study, but we can benchmark it with other studies in the same lesion lengths, more than 20 centimeter. But of course, this is only from a point of interest and for educational purposes. But if we really want to compare one with the other, if we want to compare apples with apples, then we definitely need to do a randomized controlled trial to see if DCB and BMS is as good as, for instance, DES or even bypass surgery if we are talking about lesions of 24 centimeters.